All right, here we are to talk about cell respiration. This is the process that cells use to make energy out of the food that we take in. So let's get rolling and talk about how we get the ATP necessary to do all the functions that we consider normal cell work. Now, when we look at this slide, you'll see that nice reaction right in the middle. Glucose plus 6O2 produces 6 CO2 plus 6 waters plus energy. That one reaction never happens inside the body. That is the overall reaction for a bunch of intermediate reactions that take place in order for the cell to get energy. And the cell gets energy by breaking down glucose. It's a catabolic reaction where the free energy that's created from the bonds that break, it's trapped as ATP. And remember, ATP is a high energy molecule because we have those 3 negative phosphates that are together, and they're kind of just a lot of potential energy just sitting in there. And we get to create that through the overall breakdown of the sugar inside the cell. When we talk about cellular respiration, there is 3 basic processes that are going to be happening. Glycolysis is going to be the first one that takes place, citric acid cycle comes next, and then electron transport chain. With citric acid cycle, you may also see the name Krebs cycle at some point, depending on which biology book you look at. I've heard it as both ways, so I may use that term just because that's another term I'm used to using. So if you hear that, that's what I'm talking about. Krebs cycle and citric acid cycle are the same thing. Now, all 3 of these processes are usually going to be happening kind of at the same time, so don't think of them as completely independent of each other, but as we talk about this, we'll talk about sort of a molecule of glucose going into the cell and sort of moving through all the processes one after another after another. So just sort of keep that in mind as we're Here's the basic outline, and this shows you sort of the order of what takes place and where they take place. Glycolysis is going to be our first stage, and glucose is going to get converted into pyruvate. That all happens in the cytoplasm of the cell. After glycolysis, then we have pyruvate oxidation and the citric acid cycle both happening in the matrix of the mitochondria, which, if you remember, the mitochondria is that double membrane organelle. You have the inner membrane has a bunch of folds on itself called cristae, and the matrix is a liquidy part that's in the middle of that inner membrane. After the citric acid cycle, products are created. They're going to fuel electron transport, and that's going to cause ATP to be synthesized at a really high rate, and a really high amount of ATP is synthesized, and finally you'll end up with carbon dioxide and water that are expelled from the mitochondria and sent out of the body as waste products. So glycolysis occurs inside the cytoplasm. When blood sugar levels get high inside the cell, or the cell has a high demand for energy, insulin gets released by the pancreas, and it attaches to a protein kinase receptor on the membrane of the cell. That tells the cell to take in a molecule of glucose through endocytosis. And once that molecule of glucose gets in, it's in a vesicle, it's going to get broken down and digested a little bit. And there's about 10 reactions that take place with this. And initially, to get this going, we have to put a little bit of ATP in. We actually have to put 2 molecules of ATP in. The net result, the end product, is 4 ATPs actually come out of putting 2 in. So since you put 2 in to get 4, we usually say that there's a net of 2 ATP in the process of glycolysis. So when we talk about this, our final there's 3 products to glycolysis. We get 2 pyruvate or pyruvic acid, those are kind of the same terms. We get 2 molecules of ATP, and we get 2 NADHs, which are electron carriers that are going to hold a lot of energy, and that's going to drive electron transport later. All 3 of these things are going to get used later in the cellular respiration to make sure that the cell's doing what it's supposed to be doing. When we look at glycolysis, this is sort of the chemical version of it. Again, we start with 1 molecule of glucose. There's that initial energy investment of ATP going in during the first couple steps. There's another investment of ATP coming in. And we see that this molecule got a little bit smaller here, and it's finally going to break down into 2 molecules. Do I care if you know what glyceraldehyde 3-phosphate is? Not really so much. What I do care about is these 2 molecules of pyruvate at the end. These ATPs that we got a net of 2 because we could put in 2 here and got out 4. 4 minus 2 is 2, that's our net. And then we need to know about those 2 NADHs. So again, the 3 products of glycolysis: 2 ATP, 2 NADH, and 2 molecules of pyruvate. Now, that pyruvate needs to be converted before we can move forward into the citric acid cycle and keep going forward in the electron transport chain. So pyruvate is going to get converted to something called acetyl-CoA. Acetyl-CoA is going to be the thing that tells the mitochondria, start your Krebs cycle. And when we look at the citric acid cycle or the Krebs cycle, we need that acetyl-CoA to go in, get it started, and then there's a long series of reactions that take place. Here's pyruvate oxidation. Look what else we make during pyruvate oxidation. A little bit of CO2, and we get another electron carrier. We get another NADH, which is that high potential energy molecule right there. So that's something else we're making. Now, if you remember, I only see one molecule of pyruvate here. And how many molecules do we make during glycolysis? 2 of them. So you're going to turn 2 molecules of pyruvate into 2 molecules of acetyl-CoA during pyruvate oxidation. Once you have that acetyl-CoA, then you can run through the Krebs cycle. And the Krebs cycle is this just sort of cyclical pattern where acetyl-CoA comes in and it goes through a series of chemical reactions. We start to make a lot of stuff. We start to make some electron carriers, and we start to make just a little bit of ATP, not a ton when we look at it. So here's our acetyl-CoA going into the Krebs cycle. It goes in and it starts to get converted. And as it goes down the conversion, look at this. NAD plus turns into NADH. Now, was that a loss of electron or gain of electron? Looks like it gained an electron there. So if it gained an electron, that's reduction. So this NAD plus is reduced, turned into NADH. Look what else is coming out of Krebs cycle. Carbon dioxide. That's one of our waste products that we saw in the overall reaction of cellular respiration. So carbon dioxide is coming from pyruvate oxidation and the citric acid cycle. We see more carbon dioxide here, and look at this, more NADH. This was that other electron carrier we talked about in the last section when we were talking about free energy, FADH2, and then another NADH molecule. Now, all of that came from one acetyl-CoA. And down here, we see a GDP getting converted to GTP. Hopefully you remember that GTP and ATP are pretty much analogous. They're kind of like the same thing. So from each molecule of acetyl-CoA, we're getting a few NADHs and a FADH2 plus one molecule of energy in the form of GTP or ATP, however you want to look at it. Now, after that, remember all those electron carriers that we just created during Krebs cycle, and the couple that we made in pyruvate oxidation, and the couple we made in glycolysis? Well, our cell's going to take those and it's going to use those in something called electron transport. Electron transport uses that chemiosmotic process that we talked about in the energy section. Chemiosmosis and oxidative phosphorylation provide by far the most amount of energy. So being able to get to this electron transport chain is very, very important because you're going to make a lot of energy there. And we'll talk about the advantage to getting to electron transport and using back to this right here. All of these little NAD pluses over here need to be used in order to fuel glycolysis. So that oxygen creates a huge advantage because once you have oxygen, you can make that vast amount of ATP. And if you're going to be a multicellular organism, you need a lot of energy in order to allow all your cells to do the functions that they need to do in order to maintain life. So if you don't have oxygen, the NADH electron never gets taken in and no NAD plus is created. That means no NAD plus is used to fuel the steps of glycolysis. Now, does that mean oxygen is always available to us? Well, not exactly. So we need to have some kind of a backup just in case we have to do things where we're not using oxygen or aerobic processes. Sometimes we use anaerobic processes. And if you look up your prefix, what AN before a word means, that means not or without. Anaerobic processes are a way to make small amounts of energy really quickly without oxygen. And fermentation is a process that our bodies use. Now, I put a picture of a weightlifter over here on the right because weightlifting is a really high-intensity, quick event. And when you do high-intensity, fast things like less than 20 seconds, when you're doing really high-intensity workload, that's where these anaerobic processes really come into play to create energy for your cells. So when we talk about fermentation, fermentation can happen one of two ways: lactic acid fermentation and alcoholic fermentation. Now, the big problem with fermentation, you only get 2 ATPs per molecule of glucose. Well, think about what we just said with electron transport. How many did we get there? About 32-ish. So if oxygen's not available, you can only get up to like 2 ATPs after you've gone through glycolysis. That's not really efficient, which isn't really a good thing. We needed that those aerobic processes to be able to move on a little. Now, when we compare these two types of fermentation, lactic acid fermentation, that occurs in microorganisms and complex organisms. That occurs in you and me. Alcoholic fermentation happens in yeast and plants. And big thing here is knowing the end product. Lactate or lactic acid is going to be the end product in lactic acid fermentation. Well, we haven't talked about, but if you've ever worked out really hard for a while and your muscles start to burn a little bit and you get a little bit tired, well, that's a lot of times we'll say that's lactic acid causing that fatigue. Well, why do you think lactic acid would cause a fatigue? What is that acid going to do to the overall pH of your cells? Probably going to decrease it a little bit. And if you move off optimal pH, are your cells going to be working as efficiently? Not exactly. You need time to reconvert that lactic acid back to pyruvate when oxygen becomes available. And this is where the exercise physiology stuff comes in. I get really interested in this stuff. Come by one day and stay after school and we can talk a lot about that stuff because I really enjoy talking about energy production. Alcoholic fermentation, on the other hand, well, ethyl alcohol is a product. That's what gets people drunk. That wouldn't be good if that happened in our cells. Yeast is what people use to make alcoholic beverages and make bread rise. When you put yeast in the right situation, it goes through fermentation, and that causes bread to rise and causes sugars to be converted into alcohol and all of that. So if you ever get into beer or wine making when you get older, you'll hopefully come back to this lesson and think about how fermentation works. Unfortunately, fermentation isn't efficient. That's because it doesn't use that oxygen to go into those higher levels of energy production. And since it's not very efficient, well, the organisms that were anaerobic, that didn't take advantage of using oxygen as an electron acceptor in the electron transport chain, were at a severe evolutionary disadvantage. And most of the organisms that are anaerobic are going to be really tall, one cell or maybe just a couple cells put together, because they just can't create the sheer amounts of energy it takes to manage a full-on large multicellular organism. That's what we got for today. I hope you enjoyed this, and we'll be moving on to photosynthesis next and talking about how plants make the glucose for us to use for cellular respiration. Hope you enjoyed. Have a good evening.